This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
This is Paul Verschure with our summer school again, together with Tony Prescott, co-chair of summer school, and Jon Kaas, one of our speakers. And Jon gave a talk about the evolution of the brain, and you showed this incredible overview and insight in how brains have evolved. So why is evolution, this evolutionary perspective on the brain, so essential to you?
Well, yeah, part of it I went in my talk, and that is that we have a curiosity of how we got here, what we are, who we are, and you can think of it in development, or you can think of in the religious terms, but you can also think about it in the evolutionary past of where we started and how we got here. Mm-hmm. And for a neuroscientist, I think the perspective of how our brains got to be the way they are is the most informative and interesting, given that what we are is so much different than what other species are in terms of abilities, in terms of what they do, and so on. And one of the statements I remember is, if you're criticizing someone, are you a man or a mouse? The real difference, the main important difference between a man and a mouse is the brain, I think, the things that the brain can do. Mm-hmm. Of course,
there are some slight morphological differences.
They have a body that's appropriate for the brain as well.
Right. Okay, but then you define, let's say, you identify evolution and let's say the phylogeny of brains as a phenomenon in and of itself. Right? So this is one perspective. But do you see it also contribute to, let's say, extracting these fundamental principles along which any brain is organized?
Yeah, so the same sort of thing. I picked the evolution of the human brain because it'd be the most interesting for the most number of people, but you could pick anything. How would you get to an unusual brain such as in a duck-billed platypus? How would you ever get a brain like that? Why is it organized the way it is? And it applies, if you're talking about rats, why do they have such a wonderful vibrissae system, mm-hmm. and so on? You can start asking questions of how any mammal's brain got the way it is from the early beginning.
But then if you let's take the platypus, which is sort of very close to the common ancestor also of our brain. Why is my brain not exactly like the brain of a platypus? What are the differences, and why are these differences there?
Well, first, there are some amazing similarities. And so all mammals will have this layered cortex, which is quite different from any non-mammal, and that's something we share in common. So one of the points I tried to make is that given the 6-layered cortex, it can be changed and modified in a number of ways to fulfill the needs of all kinds of different species, and depending on what they need to do. A lot of species have a high reproductive rate and a short life. And if you're going to have a short life, you need a high reproductive rate. You need to reproduce early and so on. So you can't really invest under those circumstances in a complex brain. It takes too long to build, too much energy. So we happen to be one of those lucky species that has figured out how to live a long time. And so we can have a brain that takes 20 years to really fully develop, and that's almost unique, that luxury. But it leads to a very complicated brain that a whole range of other species never invest in. They just want to get reproducing within a few months, and they're unlikely to live more than a year or two. So you have those kind of brains. But a duck-billed platypus has taken a very unusual tact, and that is that it lives in water and feeds in water, Some other animals have done that, mammals have done that, but in this case, they were able to develop from mucus glands electroreception. And at high levels of current, I guess we all have electroreception, but to get the high level of sensitivity, it's not clear exactly how that's come about, but it's happened independently in several fish and so on. And once you have that, you can exploit this environment of turbid, muddy water that you can't see in, you can't really hear when you're adapted as a mammal to be above on land part of the time. So you can touch things, but then you're already up there. So electroreception is what makes them unique and makes them different as getting electroreception. They've refined that to a tremendous degree, become their most important sense.
So the duck-billed platypus is interesting because it split from the main line of mammalian evolution a very long time ago and followed a very distinctive path. So it's almost now a unique creature. Now, the common ancestor that the duck-billed platypus has with other mammals lived, around 250 million years ago? And what is your view of what that animal was like, why it evolved to be different from a reptile, and how the evolution of that first animal changed the brain in such a dramatic way?
Well, you're asking a tough question because of the lack of intermediates. But when it was proper to talk about the ancestors of non-mammalian ancestors of mammals, they talked about them being mammal-like reptiles, and their brains were bigger, but it wasn't clear whether they had a neocortex or just a dorsal cortex, and it's not clear how dorsal cortex of a single layer of pyramidal cells with a different kind of wiring diagram and processing of information and sending information back out, how that kind of cortex changed to the neocortex. And in my talk, I emphasize neocortex because this structure is not new. A lot of people complain it shouldn't be called neocortex, and some people have called it isocortex, but it's new in the sense that it's a 6-layered structure that evolved from something that was a 1-layered structure with fiber layers on each side, so you could really call it 3 if you wanted to. But it's one layer of cells that get the input, and the same cells provide the output. So it's very simple in kind of the wiring diagram it is. Changing from that into 6 layers gave the great flexibility and possibilities to early mammals to diverge and develop in so many different ways. A lot of the ways were just simply modifying the way sensory inputs were represented, enlarging what was important, putting more neurons into what seemed to be behaviorally important. So you get large vibrissae representations, for example, or large nose representations, or large mouth representations. All these are common variations in the somatosensory system. A lot devoted to vision, little devoted to vision, these kind of things. I'd say these are what we'd say is rather modest modifications. Echolocating bats devoting a tremendous amount of space and cortex to the echo frequency. Another example.
So would it be true to say that the dorsal cortex of the stem reptiles from which these first mammals evolved was a relatively simple layered structure, just 3 layers, but was it also doing sensory processing?
I would say it had more to do with something like memory, or something like there's evidence that it had to do with habituation, which would be a kind of learning, short-term learning. And this would be very valuable, but it wouldn't be very useful in detecting a bug from a ball, or making discriminations of what kind of sensory information this was. That information would be done subcortically.
So the auditory and the tactile and the visual brain of the stem reptile would be a midbrain. And then in the first mammals, this dorsal cortex would suddenly start to take over or be involved in the representation of these modalities.
Yeah. Midbrain and parts of the forebrain that would be now considered basal ganglia in the mammal. And certainly the amygdala was a large part of this subcortical processing, and still is for a lot of kind of behaviors.
And the pressure to develop that new function for dorsal cortex, would that be to do with the lifestyle of those first mammals and how that had shifted?
Well, it's presumed that the lifestyle would have been a little precarious for them because you had all these very fast reptiles, dinosaurs all around, many of them predators. And so it was assumed that they would be nocturnal. Their skeletal features suggest that they were nocturnal. They were small. They were living on insects, or maybe some of them eating some of the infants of smaller reptiles and things like that, eggs perhaps, but mainly on insects, and scurrying around and hiding at night. And a couple things became important. One was a change in the auditory system, in which the bones of the jaw became dissociated from the jaw. So you have the three bones of the inner ear, which really defines mammals as something unique, and that allowed a transduction of high-frequency transduction of sounds for the first time. So early mammals had a nice advantage over reptiles in that they could communicate mother to offspring, or offspring could make a separation call or something at a high enough frequency that a predator couldn't find them. And they started to exploit this kind of life. And now we presume that auditory inputs to dorsal cortex, which would be now neocortex at some stage, would have emerged for the first time. And that kind of auditory processing at a higher level would emerge with the ancestors or with the first mammals.
Do you see then also a link of olfaction here? Because about nocturnal animals, so you want to sort of rely on sensory modalities that don't require light. And now you emphasize audition. Recently, there was a paper out where people looked at the fossil record, making the argument that actually this whole drive towards the cortex was very much grounded in olfaction. Do you support that?
Well, clearly the first mammals emphasized olfaction a lot, and this is a really old idea going back to Herrick. 100 years ago, there were the evidence from looking at mammals and in a comparative way, but also some of the fossil evidence suggested that olfaction was very important. They used mainly comparison of living mammals, but it's been substantiated by everything that's been learned since then. And so that if you had to look at the forebrain, it was dominated by olfactory processing. And this would be very handy for a nocturnal animal to find their food, find their mates, not use vision, not really use sound so much, because that would lead predators to find you.
So the olfaction is obviously crucial. But another interesting thing with the first mammals is the emergence of hair. And I think it's presumed that before hair could have a thermoregulation function, it would probably have had a function as a touch sensor. And of course, that was probably in the form of the whiskers, the vibrissae of the first mammals. And so then you also have somatosensory cortex. So
yeah, it becomes it's very important. The sensory hairs probably were the most important feature of early hairs, as somewhat distant, or they can touch something before they quite touch it. The longer the hair, the better. And this would be especially a great advantage in poor light or bad light, where vision wouldn't be so useful. As you'd touch something and you could back away before it was too late, or decide what kind of behavior you might initiate. And a good example of that would be in naked mole rats, which have been studied at Vanderbilt and other places. And they're not naked. They have these sensory hairs, but the other ones are gone because they don't need them for thermoregulation. and they're very driven by what hairs you touch. So they don't see you touch with a little stick or something. One part of the body, they go forward. Another part of the body, they go backwards, because where they're touched determines what direction they're going to move in.
So now we have sort of a sketch of this early mammal, but it also then had to develop a brain that would match these capabilities, and then the idea would be it would contribute from this step towards a 6th-layer cortex, right? It would just sort of highlight the isocortex in this case. But still, then within that, so now there would be two questions at least. On the one hand is, okay, how do I get from a 1-layer sheet of cells performing some very simple, possibly memory function or learning function, to a 6-layer structure, and also so fairly rapidly. But then how do I make that match all these variations on morphology, on mixing of different sensor modalities efficiently and rapidly? Because we're talking Cayman explosion roughly, so we have all these different types of animals emerging. So how do you see the 6-layer cortical sheet being rapidly adjusted in its details to then the specific requirements? So how do we get from 1 layer to 6? And then once we have 6, how do we tune it actually to the specifics of the specific organism in which you want to have that cortex working?
Well, the speculation of how you get to more layers would be that the original organization really corresponded to layer 5 pyramidal cells. They get inputs on their apical dendrites, and they send outputs subcortically, and this is what dorsal cortex does with a few interneurons. How those other layers came about and evolved is really rather uncertain. People working on development probably will come up with, and are coming up with, some good suggestions about cell migration, cell division, changing the cell replication cycle, and so on. But it's really uncertain because we don't have examples of intermediate forms. Once you get to the 6 layers, you can modify the sensory representations that are there, as I already mentioned, in all kinds of ways. But the real powerful advantage of cortex is that you can start to replicate cortical areas and increase the numbers. One idea that we talked about a long time ago is just you duplicate areas that are already there and then modify them differently for different functions. I don't know if this is the way new areas emerge or where they emerge gradually by internal differentiation. But the idea of duplicating is something that's an old advantage in evolution, where you duplicate body parts. And as you can see in segmented creatures that have more segments and more body parts that were originally all alike, and then you start specializing them as in a lobster with different of the appendages modified for different functions. And you can think of cortex with more areas doing something similar, is that you're now modifying different parts that might have been quite similar in anatomy and connections and making them more and more specialized for different kinds of functions. If you don't have a number of areas, you can't specialize for different functions with one area very easily. You can subdivide an area into more layers and get some of that in a single structure by having layer differences. But really the best way to add is to get more areas and then specialize them. And you specialize them in terms of what their inputs and outputs are, but you specialize them also in adjusting them to what they're best at doing or what you want them to do. So you can have dendritic arbors that are large or small, and they collect information from a few neurons or a lot of different other neurons. You can use different neurotransmitters. You can have different neuromodulators that will affect them in a different way. You can, and you can end up, as we hear in all these talks, with neurons that are sensitive to very particular kinds of combinations of sensory information.
Yeah, but there's, in my mind, there's sort of a gap still in that answer, because, well, if while I'm changing my morphology, it's not that I can just change my morphology and I flop around for a few generations, and at some point in time I have a brain that matches my morphology. These changes have to happen in a very coordinated fashion, right? So that means I would hope that you have an answer just for my own to satisfy my own ignorance. That would mean something like, well, there is a very specific genetic control that allows you to change body parts and in some sense change also matching neural structures in some coordinated fashion.
Yes, absolutely. So one thing that you can imagine is that you change brain parts without changing peripheral morphology at all and get more out of the kind of information that's coming in. But there is a cascading sort of modification that occurs in any system. So if you change one part, the whole system is changed by that, it's, at least in development, but also in mature animals. And that's due to the plasticity or the flexibility of the system to always adjust to change. And so one of the major mechanisms of evolution is that if you change the sensory inputs, say you change the cochlea so that you can now send signals at higher frequencies, the whole system, if you did that in any animal that didn't hear high frequencies, if you gave them the possibility of high frequencies, the whole system in development would adjust to that and have neural space devoted to this. If you doubled the number of receptors that come from the face, the whole system would adjust to that, and you would have more processing throughout. And
so your answer is essentially to say, look, the first trick was maybe to develop a brain or a cortex, if you want, that is, let's say, hyperplastic or hyperadaptive. And then after that, you can start to play with your morphology to sort of see how you can optimize yourself to a specific niche.
Yeah, you have things that will happen automatically, and one of the papers that from van der Loos is how the periphery instructs the brain how to build itself, and I think that paper was not as influential as it could have been, in part because he was looking for genetic reasons as well at the same time, and he diluted his own message that way. The powerful thing was working with mice. If they're born with an extra whisker or a whisker short or a couple whiskers extra or a couple short, you still have a whole vibrissae system that incorporates those new information or that lack of information into the number of barrels or barrelettes or whatever in different parts of the somatosensory system. So the system developed to accommodate the change in the periphery. And even that change in the periphery is somewhat indirect because it depends a bit on how folds in the face occur in development, and that determines how many whiskers there's going to be.
So we have this early mammal brain, which is plastic and allows quite a lot of diversity to happen, but it's still really quite a small brain. And then you mentioned in your talk something that was very interesting, that around 60 million years ago, the reptiles disappeared, and that this was an opportunity for a new radiation of mammals to diversify into a whole new set of niches in which they could live. And that there were then some really quite dramatic changes in the brain as a result of that for some groups of mammals. And one of those, obviously, is the group of mammals that led towards primates and eventually to ourselves. So what do you see as the most important changes from those earlier mammals to, say, the early primates?
Well, it did give an opportunity for animals to become diurnal. It became a lot of opportunities for different kinds of environments that they could occupy and so on. Their risk of being preyed upon changed from these very efficient reptilian predators to the mammals that had to evolve into becoming efficient predators and opening up that predator environment for them. But early primates developed apparently from animals that were emphasizing vision more and living in bushes or trees and trying to go into the pine branch environment and live on insects and find food there. And it gave them an advantage of escaping some predators by being away from them. but it put demands on visual processing in dim light that were very powerful, and so they had to devote more to vision. So eyes were big, a lot of receptors, but the whole visual system started to expand and devote more neurons and more cortical areas for sequential processing. And already in the ancestors of present-day primates, the temporal lobe would have been expansive. The fossil record shows the temporal lobe and occipital region are both expansive. Both of them are thought to be completely devoted to vision. And so the cortex now is going over and covering the whole midbrain and much of the cerebellum. You can get that from the fossil record. And of course, if you're going around and catching things rapidly in fine branches, insects that could hide or escape or defend themselves in some sense, by you have to be agile. You have to be able to hold on to a branch that may be moving up and down and reach and get something that may be on another branch and isn't moving or is moving in a different way. And you have to use visual guidance for these kind of movements. And grasping things with a forepaw would be something to be important and new, sort of, instead of grabbing things with your mouth, grabbing them with a forepaw. So eye-hand coordination would start to become important. And so all primates then, all living primates, have a huge investment at the cortical level in processing visual information, but also in processing sensory information relative to simple motor skills.
So and I think that's quite important, the idea of the hand, and also the hand being away from the mouth, so you can look where you want to eat something or pursue something and go with your hand for it. And in a way also that frees you from having the need for the whiskers quite so much, because you now use your hand as this organ for touching further away in the world. But one of the things about primates, I think, that people notice is that they live in large social groups, and that the development of these societies of primates was possibly one of the important factors in their success. And is that reflected in the changes in the primate brain?
Not all primates live in social groups, and a number of the prosimians don't. So it's uncertain, I think, if the first primates were social or not. There'd have to be some kind of evidence one way or the other, and I'm not aware of any. They might have been solitary and nocturnal. The suggestion would be that they're nocturnal. But if you're going to go to the ground, or you're going to be out in the daylight and the ground, it's a tremendous advantage to be in a social group, because then you can be detected visually by a lot of different predators. You need a lot of sentinels. So you have to have somebody warning you that there's danger, and you're not too distracted by trying to find food or other things. So a social group then becomes very important. And a side benefit of a social group is you can defend a territory against other social groups or non-social animals and drive them out.
And that would then impact on the sort of non-motor or less motor and less sensory areas of the brain, and the way that they would change.
This would impact on, I think, especially frontal cortex and enlarging frontal cortex. And you see in the monkeys and all the anthropoid primates that the frontal lobe is really well developed, but it's not very well developed in the prosimian primates by comparison to monkeys. By comparison to most other animals, the frontal lobes are quite well developed in even prosimian primates.
So now we went with our cortex from the first mammals, the first vertebrates, up now to the primates. But in some sense, that's not the only part of the brain that evolved or that changed, right? There's, let's say, also an underlying architecture. And also in your talk, you mentioned the MacLean proposal of the triune brain, which might be considered a bit naive. But it was like one of these attempts to try to say, like, okay, but these are the different parts of, let's say, an overall neural architecture that we have to consider when we think about this evolutionary perspective. So what's your view on then these key ingredients of that architecture that actually leads to a successful brain?
Well, if every mammal, every animal has a successful brain in some sense. It has to be successful or they wouldn't be here.
They want to share features. That's the point, right?
But you're right in pointing out that it's just not neocortex, but the whole brain is being modified in each line depending on what their requirements are. But they interact. And give a couple examples. One example, of course, is optic tectum or superior colliculus, as you're talking about mammals. You don't get a cortical input to the optic tectum in non-mammals, but you get a cortical input to the tectum. But the regions that project to the cortex or the tectum, superior colliculus in mammals, is variable across mammals. So if the inputs from frontal motor areas, for example, frontal eye fields and so on, that's unique to primates and maybe a few independently involved in a few other animals, such as some carnivores and so on that are highly visual. So you're modifying a structure that has been important for vision through all vertebrates, basically. But you're modifying it by adding complexity to it and adding other ways of influencing its functions.
But do you see this adding of complexity mainly occurring at this level of this isocortex, or is it then also matched by the added addition of complexity at these supporting midbrain and lower structures?
So the midbrain will be modified, and architectonically it's modified. You can see it's changed greatly in different lines in terms of architectonic complexity. So its functions will be changed as well. Mm-hmm. The important feedback into the thalamus is very old. That there's a projection from the optic tectum into the thalamus, but how that's distributed to cortex has changed as cortex has changed. Its downstream projections become more important because they're influenced by other inputs, and so they're not really doing the same thing as they did before. Mm-hmm. And that you could think of almost any structure. The amygdala would be responsive to direct inputs from the thalamic inputs to the amygdala that wouldn't depend on cortex at all, and they're highly important in a lot of mammals, but certainly non-mammals.
But do you see, let's say that because in some sense the sort of the simple view on the phylogeny of also the human brain would be like, okay, you have all these subcortical structures, and they were sort of a constant, and then this magnificent isocortex grew on top of that, and that added all these amazing functionalities. An alternative view would be to say, no, actually, it's a bit like we discussed earlier. Morphology and brain has to match, and in some sense you could say, okay, isocortex and its and subcortical structures have to match as well. So it's always a co-development and change in these structures, as opposed to these subcortical structures remaining constant and then your isocortex exploding. Mm-hmm. So where do you position yourself in that debate? How should we look upon that?
Well, you're talking about systems and include cortical parts and other parts of the forebrain and parts of midbrain, hindbrain, spinal cord, and they all have to be coordinated and interrelated. But a lot of what happens depends on the changes in cortex to sort of drive these other changes, in my view. As motor centers in the cortex become important for controlling digit movements and hand and so on, you have to modify the circuitry in the cervical spinal cord that will deal with this control of muscles. But deciding when to do something or how to do something and so on would depend on cortex. So it would be a series of gradual changes, I think, over many, many generations that would modify different parts, but always together, so that it would be pointless to have some function you couldn't use.
Okay. But then if we now reduce a bit the level of granularity in that discussion. Yeah. So now we look at, let's say, this brain with its different parts. We might have to sort of define what the key subdivisions are there, co-evolving with the morphology. But now these larger chunks, like isocortex and its underlying structure, like thalamus or basal ganglia or amygdala, again consist of the subcircuits. And these subcircuits also will have some invariant features and also some variable features. So what's your view on that? What are the invariant features of such an isocortex, and what are the variable features as such a brain is on one hand changing phylogenetically, but also changing across the different modalities where it has to deal with?
Well, tough question, but I've tried to emphasize some of the things that all mammals would have. It seems unlikely that we would evolve any mammal without a somatosensory system that had somatosensory cortex involved in it. There are several divisions, but at least one division, because we depend on this sort of sensory information so clearly. And once you relegated it to the cortical level, it seems like you're unlikely to change that and put it and do those functions some other place. I can imagine the visual structures completely being lost in animals that no longer have functional form vision, as we see that those systems can be greatly reduced. Auditory could be lost. I can imagine it's not so important in some animals that live underground in tunnels, for example. You know what direction the sound is coming from because your body is blocking it or not and muffling. And so sound localization is reduced. Sound is still important. You could imagine things being lost. In terms of higher order functions, and it's completely, you could go in any direction, I think. It could be modified in any way. It'd be easy for midbrain structures such as the superior colliculus to become completely auditory or completely somatosensory in function and not to use vision at all anymore. It is already multimodal, and so you could just change functions. But the motor functions might be always very similar towards orienting towards a stimulus or something like that. Relay of sensory information as another route to cortex might be completely used in a quite different way.
Right. So, John, a little procedure-wise.
Yeah.
So Tony wants to discuss you a little bit developments in certain domains of science, like comparative neuroscience and so on. But then he has another appointment. So then after that, I want to go back to some more specific questions, and then later we edit the interview that sort of these concluding questions go towards the end. So that you're not sort of shocked by Tony leaving.
And if you want to stop for lunch, then tell him, because otherwise he'll talk to you till it gets dark.
Oh, wait.
Exactly.
I will have to go get my wife for lunch pretty soon.
Exactly.
Sometime.
Sure.
Okay. So, a couple of things. The first thing, I wanted to ask about multisensory areas in cortex. So it's clear that these early mammals have these new areas, or sort of radically more sophisticated areas for analyses of the auditory and visual and somatosensory inputs. Now, at what stage and whereabouts in their brains do they fuse these inputs? And how does the multisensory system evolve as mammals diversify?
Opinions on this have changed a lot in recent times. When we were working early on in mapping visual areas 40 years ago, it was popular, and we believed it, to talk about visual areas or auditory areas or somatosensory areas. And we said, most of the processing is done within a modality, and only at the very end, when things are very sophisticated, do you start to integrate modalities. And this partly was due to the methods of recording from anesthetized animals, where you really have dampened the responsiveness of neurons tremendously. And so you're getting this dominant input and say, yes, that's visual. And now when the anatomical methods and recording methods have been improved, you'll start to see people saying it's multisensory everywhere. And Barry Stein had a book, is the editor of a book a few years ago on multisensory systems. And you could say then it's amazing how fast multisensory processing evolved, because it wasn't there very long ago, and now it's everywhere. But it depends on what you're looking at. Clearly, people are talking about in primates that primary visual cortex gets auditory input, maybe somatosensory input, maybe not so directly, but some inputs rather directly. If you look at a small brain animal like a rat or a mouse, you're seeing these kind of connections. Primary visual cortex will connect it everywhere almost. You're getting, and if you look carefully at striped cortex projections like Henry Kennedy has been doing, and 99% of the connections can be accounted for with maybe 5 areas that you're talking about. But that 1% probably goes to 15 other areas and has some influence. So I think it's hard to find a neuron in the cortex that's not influenced by more than one modality. But whether it's driven by more than one modality, that would reduce the number of places that you would find that. To give an example within a modality, you will find in primary somatosensory cortex representing the hand in a monkey, neurons with small excitatory receptor fields on different parts of the digits, rather small, responding nicely. But if you get the neuron responding, and even in an anesthetized animal, and now have a second somatosensory input somewhere else on the hand, it'll influence that firing. Then you go to the other hand on the other, and you will also influence that firing. And there's practically very few direct connections between the two hemispheres. For the hand areas, there's more connections would be from higher areas and then feedback connections. So it's not clear exactly how this is done, but it is clear that our traditional pictures of what neurons are responding to has been greatly constrained by our methods of looking. And when you look in other ways, you'll see the neurons are influenced by much more outside what's they call the classical receptive field in their own modality, but also from other modalities.
Well, this is, I think, very interesting. So there's a caution here for students that if you're reading the older literature, you may want to make sure that the opinions on these things haven't changed, because there's been some really radical changes in methodology here that have made us revise our views on some of these fundamental questions. I guess another thing would be to say that this question of the multisensory cortex is really still open, that there's a lot more we need to know about this.
And there are recognized areas in primates and that are where neurons respond very readily, even in anesthetized animals, to auditory and vision, or vision and tactile and so on. And these would be called, in earlier times or present times, multisensory. Some of these areas have been called multisensory for a long time because of these responses. But 50 years ago, all the recordings were from a few primary sensory areas, because anesthetics used at that time just blocked everything else. So you had no chance of really talking about multisensory.
So going to the field and how it's developed. So you've been in this field for a long time, and you, I think you were lucky to come into it at a time when people had this strong interest in comparative approaches and looking at different brains and different species. And now we have these fantastic new range of techniques to do this comparative study. But how do you feel that the field of comparative neuroscience is looking in correspondence to the rest of the field? I mean, is this work happening, and where would you like the focus to be?
Well, first, it's true I've been in the field for a long time. And this year I got a letter from Science magazine, where we like to publish every once in a while. It's getting harder. And they said, you get a free subscription from now on. You don't have to pay for it anymore because you have subscribed for 50 years. So there are some advantages. I would say not that many, but there are some. And your question now is, how do I see the changes that have happened in comparative studies of brains? How has this field been influenced by technological advances as much as other parts of neuroscience?
It's how you see the future for comparative neuroscience, really more generally. So there's obviously still many questions to be asked here, and how should we go about it? How should we balance our resources as a community between this comparative approach that you've been exploring and other approaches that are maybe focused more towards single model animals?
Well, I think the model animal approach has been a good one, and I wouldn't disparage it. Because if you work on a particular animal, you gain a lot of information that others have developed for you. If you work to another model or another animal, you have to do a lot of things over just to get to the point where you can answer the next question. So that becomes a problem. And sadly, in some ways, I started out working on cats as the model system, and they've been largely abandoned as a model system. And there's a tremendous amount of information known about brain organization in cats from years and years and years of study that is in some sense now not used to its full capacity because people have moved on to other models. That said, I think that we should always work on some primate models and some rodent models and maybe a few others, simple models. But simple models are limited in many ways. There was a debate at the Vision Science meeting, a friendly one just for the fun of it. And Tony Moshman was going to defend using simple models. He works on monkeys, so he was picked to defend using simple models. And he said, why stop at the mouse? C. elegans would be perfect because they're so cheap. The cost per individual is not even worth mentioning. They're so cheap. And they can do so many things. You can train them to move, turn to the left in a maze and all kinds of things. And he went on for about 15 minutes about their virtues. But he said there are two problems for vision research. They have no eyes and they have no brain. So you have to have a model that has the capability or the potential of answering the kind of questions you're interested in. And that means that we need a range of models because you can answer very general questions or very specific ones depending on what model you're using. But we also have a chance to study now a whole range of species and do it very productively because the methods are so powerful. You can learn so much so you could take a new species, something that's never been looked at before, and you could know a lot about the brain of that species from one laboratory in 3 or 4 years. I think you could come up close to having a basic understanding of the brain organization in a relatively short time because the methods are so powerful. And they were so poor when I was starting out. You were so limited in what you could do, and they were worse before then. So and that people came up with such good ideas early on such limited information is totally amazing.
And given your vast experience and knowledge of different mammalian brains, are there particular animals that you think we should be exploring more?
Well, from the point of view of evolution, brain evolution, which is of particular interest to me, it's important to learn more about the relatives of primates. Also, how much variability is in the primate order. We know a lot about a few monkeys. We don't know much about the diversity in monkeys. We know very little about apes. We're learning quite a bit about humans now from non-invasive methods. But so there are big gaps that haven't been studied very well. Primate studies aren't very frequent. A review of publications over the last 15 or so years by Paul Manger showed that almost all the studies are on mice or rats or humans. Takes almost all the neuroscience studies. Then out of primate studies that aren't human, then you have macaque monkeys studied. And then the next would be prosimian galagos. And they're only studied in 2 laboratories that I know of, so a single person or a few people could make a big difference by filling in gaps in particular areas. It doesn't take many, but we have a lot of gaps. I think to study animals like the monotremes would be very important because they're so unusual. We don't know anything about the brains, practically nothing about any of the larger marsupial. We know a lot about opossums because they're available in this country. In South America, they're small. You can get them in the laboratory, but we don't know anything about the red kangaroo, giant kangaroo brain, how it's similar, how it's different. We don't even know very much about carnivores at different sizes. So you could pick almost in any order animals that are just a mystery. And someone studying them would be bound to find something interesting and informative just by falling in and trying to do this. So one of the things that could be done in a lot of different countries, and there's been a tendency to do this in Brazil, take the native animals that are there and try to understand their brain organization. Yeah. So they're looking at, say, the very large rodents and so on, and how their brains are organized.
So then, to follow up on that, so you also showed in your talk this an earlier map of the cortex, and most of it was actually white, right? So by now, how much of this map of the cortex do you feel have you really filled in? Can we say, look, these are areas where we really have understanding how they're organized, how structure maps to function, and how big are the gaps?
There are major gaps, but depending on who you ask, people will feel, well, we've got the gaps pretty well filled in or not. So you can look at Brodmann's maps. There are no gaps.
Right.
But there are a lot of errors. Mm-hmm. And different kinds of errors. We can make complete maps at any time, and it depends on a tolerance of ambiguity, or we can say, well, we're basing this on a very limited amount of evidence, and so on. And the kind of map that, for example, that Fellman and Van Essen did had a lot of uncertainty, which they recognized and talked about. And I think it's important to talk about uncertainty so people don't get the impression that we really understand all the little divisions and subdivisions of the brain and where they are and how they're organized and how they interact.
Mm-hmm. But if you would have to give this a number, would you say, look, 20% is reasonably well explored, so uncertainty is low for these areas, let's say primary visual, primary motor. There we know what we're talking about. Association areas is, let's say, synonymous with saying we have no clue.
So out of the proposed about 100 areas in a macaque brain, I would say we probably have a good understanding of about 20. So that'd be about 20% in that brain. But it depends on what you mean. I would say that you can find good evidence for a functionally distinct region, but to define it precisely in terms of boundaries becomes a real problem. So if we talked about VIP today, in the talk today, people are very uncertain about what you're talking about. You know roughly where you are, and you say, if I'm in this region, it must be VIP. But it's hard to pin down exactly where the area is, what the boundaries are. Is it variable across? Are the boundaries variable across different individuals, and so on? So I'd say that's a subdivision of the brain that's reasonably well understood, but it's not understood to the extent that you can say, I know I'm in it, or I don't. I'm not sure whether I'm in it. Those kind of questions. you don't know what all the connections are because we don't know how to define the area precisely.
Right. But then, in some sense, also in your recent work, I think this also illustrated maybe this issue a little bit, because if you look at motor areas, then in some sense, the standard knowledge would be that you have a fairly abstract representation of, let's say, the direction of movements, acceleration of movements. We're close to the kinematics of the whole of the body that we're then controlling. But in your recent work, you show that if you stimulate in these motor-related areas, you actually can get whole coordinated movement pattern. So how do you then link or compare that to our, let's say, standard understanding of these areas we thought we knew? You say, okay, there's something like a population response that gives you some kinematic control. But now it seems that in your recent results, you're saying, look, actually we're talking about highly coordinated control of stereotype behavioral patterns. So where are we going with that?
Well, motor cortex, even primary motor cortex, is especially interesting because the early maps didn't quite give the details of the way that they're organized. So you'll have a hand area, you'll have a foot area, a face area, tongue area, if you even. But within the hand area, you'll get multiple representation of the digits movements, the same digits. You'll get a digit movement that the very next site where you stimulate it next to it, you'll get a wrist movement. Or you might even get movement at the elbow, or you might get, and the same digit movement would be paired with other digit movements, or just that single digit movement. But that would be repeated over a large region of cortex. You would find the same movement again, and this seems to be different than some of the sensory representations. Why are you repeating these things? And you can say, well, it's important because any particular movement might be conjoined with any other kind of movement, and you want to have them close and interconnected. I think now that the long-term stimulation is showing more purposeful behaviors or movements can be elicited from primary motor cortex or premotor cortex, is starting to give another perspective on this issue. Part of a cortical area of M1, of the hand area, will be involved in some kind of behavior with the hand. Another part will be involved in another kind of behavior with the hand and joined with arm movements and so on, depending on what it is. And so this adds a complexity as if parts of the area are working somewhat independently from other parts of the hand area. You're not involving the whole hand area in one task. You're involving part of it, and other parts will be involved in other tasks. That's what seems to be suggested by these kind of stimulation experiments. So it's more complicated than it seemed originally.
Right. So are you saying with that in these motor areas, you would have like a library of discrete behaviors that are then sort of biased with respect to the limbs that are being involved in these behaviors? Should I look at it like that?
I think you can look at it that there's an organization, a gross organization of foot to tongue from medial to lateral. And then within that, there's a more complex organization. But that organization is similar across individuals of the same species and even across members of the same family, different species. So you have this preserved. And the reason it's preserved must be that it in some way is specified during development to come out in a particular way every time. On top of that, you must have tremendous amount of being able to modify these circuits by experience and training so that you not only want to be able to do some tasks that every animal has to do, every person has to do, pick up something, but you want to be able to develop specific skills that for a human, maybe making pottery or something, like you practice and you get good at it, but it could be anything that you practice and get good at. For many species, practice and modifying the system may not be that important. If your average lifespan is 8 months, you don't want to spend a lot of time modifying it. But for a long-lived species, this modification can be very important. So our motor cortex part of the motor system is greatly expanded. It's got all these premotor areas and cingulate motor areas in it. So there's a tremendous amount of cortex involved. And exactly what this means isn't so clear, but there's a big investment in motor control at the cortical level that interacts with posterior parietal cortex, frontal cortex, sensory inputs of various sorts.
Right. But do you see these behavioral primitives now? So we're not talking motor primitives, we're talking behavioral primitives matching the ones that are represented and controlled at subcortical levels, like in areas like the central gray, for instance. Do you see some matching there between these primitives? That are then, so that means the motor cortex provides you an interface to these subcortical areas on which you can then start to play using, let's say, your planning and learning mechanisms you have at cortex. Or should I look at this differently?
No, I think you're looking at it in a good way, because it's has been unpopular to talk about primitives or built-in sort of things in a human brain, for example, and say it's all learning or almost all learning. But then if you start looking at mammals in general, you see that there's so many things that would be too costly to learn, costly in that you wouldn't live long enough to learn them if you didn't know how to run away or escape or recognize a dangerous situation, or to find your mother, or whatever. There could be so many different things. There might be very rapid learning in the kinds of imprinting for many things that would allow a pre-adapted system to rapidly congeal into eliciting a kind of behavior. That's hard to rule that out. But then you have a whole system of levels of say, a sensory motor system that would have to be working in concert, pre-adapted for certain primitives, modifiable, hopefully in some ways, and on top of that, then completely learned and modifiable sorts of motor behaviors that would be unique to the individual if they bothered to learn them.
Right. So, John, to get to the finish line with our discussion, two questions. So as you already indicated yourself, you got awarded this free subscription now to Science because of
yes.
We have been paying enough money to this.
I haven't gotten the first issue free yet, though.
Exactly.
We'll see.
So, based on this broad experience you have in the study of brains and this comparative study of brains, what would you now see as the law of Jon Kaas in our attempts to understand the brain?
A law?
Yeah.
I'm not sure what a law in we used to talk about laws in science quite a bit, but I not so much anymore. So
this is your chance.
Yeah. So I could make a law.
Yeah.
John Ullman and I, when we started defining visual areas, thought maybe they'll name a visual area after us sometime, but it never happened.
Or an asteroid, maybe.
So now is a chance for a law.
Yeah, exactly.
I don't think I can think of something that would fit the law. What I would like to take from other people that have made studies in development is that one level of the system will specify what the next level, how a lot of its organization, so that you have this chain naturally that starts with sensory inputs and modifies the system to adjust to whatever the sensory inputs happen to be. This principle probably works anywhere in the nervous system. You make a change at any level in a complex network and development, and the rest of the system will develop to accommodate that. And this makes brain evolution and change fantastically more easy, because you can imagine if it's in some sense based on genetic change, that you wouldn't want something that you had to change the genes at 10 different places, or the gene expression at 10 different places to get what you wanted. You would like to be able to make a change one place by some fortuitous accident of genetics. And that dies out because it didn't work well or it's propagated. But for it to work at all, it has to propagate itself through the whole system.
Mm-hmm. So the Jon Kaas law would be that when we look at the brain, its power is actually its ability to adjust to many different kinds of sensory inputs, also across different morphologies, right? So it's very much a very general design of many possible brains as opposed to a single one.
Yes.
And that the driving force is really how am I changing the inputs to this hyper, let's say, plastic control system. Yeah. But I don't know how we can summarize it in 3 words. So then the second question would be, if we're going to get you back here 5 years from now, and we do a similar interview, I want to ask you, okay, did your prediction work out or not? So what's the one prediction you would like to make today that you're most enthusiastic about, that you have the strongest confidence in?
I think that we're going to have, at the cellular level, a much better understanding of a whole range of brains. And one of the surprises that's coming out of looking at just counting neurons is a whole change from views that were made 20 years ago. And the view then was that all cortical areas are basically the same in the sense that their hardware is the same. Their neurons are the same. If you run a pin down through cortex, you'll count the same number of neurons no matter where you are and so on. The variability is turning out to be really tremendous, and it's possible to look at different aspects of that variability now. And one thing that's true, that's going back to the early studies, they said the one exception is primary visual cortex in primates, that the number of neurons is twice as many per unit volume of tissue than other places. It turns out that that statement is absolutely true for humans and for monkeys. It's less true for prosimian primates. It's less true for New World monkeys and Old World monkeys and so on. So it's variable for primary visual cortex, but it's also variable in many other areas. And we don't know completely for humans yet, but macaque monkeys have variable numbers of neurons or neuron densities for different cortical areas. So they've specialized by changing the density of neurons, which implies changing the sizes of neurons, so bigger or smaller, because that affects the density that you can pack them. And that wasn't appreciated, and it's just starting to become known as a big variable. But this variable is is a major variable in macaque monkeys. It's going to turn out, I predict, to be a major variable in the human brain, and it's not going to be a major variable for most mammals. The neuron densities, the neuron shapes, and neuron functions are going to be more standard for most mammals, including prosimian primates.
Okay, great. Well, Jon Kaas, thank you very much for this conversation.
Okay.
That was great, Jon. Thank you.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.